Dr. Tore Skotland, Nanoparticles for Clinical Use, Importance of Degradation and Excretion. Please.
So, how do I move this? Is that one?
That's correct.
Yes, thank you. What I'm going to tell you is heavily influenced with my background. I have been working 26 years in pharmaceutical industry and heading work with metabolism, excretion, biodistribution of all type of contrast agent in the main biomedical pharmaceutical industry. Nycomed, based in Oslo, was bought by Amersham and then bought by GE Healthcare. So I've been involved in bringing 5 products to the market and another 5 into clinical trials. I decided to leave industry in 2009, and I'm now working in the project group of the chair of this session. So some of the messages. I think there are a lot of very interesting nanoparticles being synthesized, but I have to add that the biological studies are often in a quality where the conclusions drawn are not supported by the data shown. And more cross-functional collaboration is required in order to proceed in an optimal way. A major part of my story will be that different type of nanoparticles, there are very large differences in studies needed, the risk of failure, and the cost of development if you want to bring them to the clinic. And we have discussed that in the review article in 2014. So, by injecting nanoparticles intravenously into blood, they have a very restricted ability to exit blood. They can pass through small holes or fenestra or liver, spleen, bone marrow in the so-called reticuloendothelial system, RES. They may pass through leaker endothelium in inflammation areas and in solid tumors. And if they are very small, they may be excreted through kidneys. So this paper from 2007, they studied in detail how fast particles with different hydrodynamic diameter can be excreted into urine. And you see here how this drops when it's increasing hydrodynamic diameter. So these data, you see that they often are interpreted as nanoparticles are rapidly excreted in urine if being approximately of 5 nanometer. I don't think that's a good interpretation of these data. In my opinion, it's much better to say that if nanoparticles are approximately 5, 6 nanometer, you can expect approximately 50–60% to be rapidly excreted. But if there are 9 or 10, practically nothing are excreted. And this is very important because most nanoparticles we aim to inject are larger than 5 nanometer. And this makes it very important that they are metabolized and excreted. That's very important both for safety issues and for regulatory issues. If your particles are based on endogenous substances, for example, made of lipids or albumin-based, this may not be a problem. And for example, iron oxide particles have been on the market as MR contrast agent for more than 20 years. But there are very little data available for degradation of other nanoparticles. Speaking about targeting of nanoparticles, by passive targeting into RES or taken up into tumors by the so-called enhanced permeability and retention effect, there are not many data from clinical studies of how much you can expect of particles to home into tumors based on such mechanisms. In fact, the only study I am aware of from clinical studies was a study with using pegylated liposomes where people tried to simulate what happens with doxil or calyx by including radioactively labeled substance, and ended up by stating that 0.5 to 3.5% of the injected dose ended up in solid tumors. What about active targeting to receptor cells? Well, first of all, I would like to make clear that we are not speaking about target-seeking particles. You can often see that in literature. People think about this as target-seeking. They are, of course, not target-seeking. I think it would be much better to call this principle selective retention. But how much of the injected dose is ending up in the target? Well, from my area of research with imaging, it was well known 20, 30 years ago that you can expect a very small dose, percent of the injected dose, to end up in the target of tumors. But even if it's very small percent of the injected dose, it can still be very important to increase the uptake and to give an important therapeutic effect. But it's important to discriminate between this therapeutic effect and the percent of the injected dose that is taken up. I would also like to add that you can often see studies claiming that by adding a targeting molecule, you have a certain increase in the uptake into tumors. But these studies most often lack the necessary controls. So it could equally well be not a targeting principle that is giving this increase. It can just be an increase of surface properties like charge or hydrophobicity. So what is, I think, is important to think about, that you must expect that much less than 5% of your injected dose is ending up in the target. That means then more than 95% of the injected dose is present in other parts of the body. And then it's, of course, very important to think about this when you think about safety studies. Most of the injected dose is ending up in other areas than the target. Some words about documentation needed for market approval of drugs. The physiochemical characterization, you need to characterize size, charge, chemical composition, and so on for all ingredients in your product. And you need to document the reproducibility of the production process. And accordingly, by increasing the complexity of your product, you can expect higher cost and higher risk of failure. When it comes to toxicity study, animal studies, I'm not going into detail, but if you have particles that are slowly degraded, you need to perform these studies for a longer time. And it's very difficult to give some very clear answer to how long you need to perform such studies. Guidelines exist for this, but they are nothing more than guidelines. You need to discuss with the authorities in each specific case. Also, when coming to other type of studies, I will not go into detail, but just from the type of research I have been mainly working with, distribution, metabolism, and excretion, you need to perform a lot of analysis. And if you then have very complicated particles, you need to do a lot of analysis. How much data the authorities will ask for, I don't know. I think they don't know. You will not get a general answer. It has to be discussed in each specific case. What is clear is that it costs a lot to bring products to the market, whether it's an imaging agent or a therapeutics. And for nanoparticles, to my knowledge, no such numbers are available, and the only products that so far have reached the market is based on lysosomes or albumin-based nanoparticles, what you can call for endogenous substances, whereas you must expect higher cost if you are going for a product with non-endogenous substances. What is very clear, more constituents means more analysis, more studies, and higher cost. And it's a great difference between trying to get approval for a non-degradable versus a degradable nanoparticles. If you want to contact a pharmaceutical company and try to ask them to develop your product further, you must expect that they will perform a risk-benefit evaluation of your proposal. I guess that all companies have more or less their own list they will follow, but you can be quite sure that they will think about what's the market potential. What can we obtain with this product? Is it a better therapeutic effect or less adverse effects? What's the expected cost and time for development of the marketing? What's the risk of failure? And especially at the step where they already have put large investment into that development. What the risk of not satisfying formal regulatory demands? And there I'm especially thinking about such issues as, are you able to make reproducible batches? What's the expected manufacturing cost versus market price? And not at least, a question that is almost impossible to be sure about the answer, but nevertheless, the companies will think about what is the risk that other companies coming to market with an equal, better, or cheaper product. Some few words about Theranostics. That's by combining imaging or therapeutic issues. There are no doubt that that way of making products is very useful for in vitro studies and animal studies. When it comes to the clinic, I'm far from that sure that it's a good way to go. It's more expensive and complicated to manufacture. Drugs must be approved for each separate use. The optimal properties are most likely not the same for therapeutics and imaging agent. For an imaging agent, you would like the product to be rapidly excreted to remove the signal from the surrounding of the diseased area, whereas for the therapeutics, you would like to circulate for a longer time to have more drugs to enter the diseased area. So in my opinion, for clinicals, I think it's better to focus upon one agent for one specific use. And I often would like to comment on these issues by saying that if I should try to summarize my experience from 26 years in pharmaceutical development, it is make products as simple as possible. Finally, as also mentioned by previous speakers, it's very important to have cross-functional collaboration with experts in all different areas from synthesis, characterization, cell studies, animal studies, thinking about using the right models, and safety studies, and also clinical studies involving people knowing the clinical need. And as also mentioned by the chair, we have a poster describing such a project just outside the nearby auditorium. So thank you for your attention.
Okay, thank you very much. We'll take one question.
I actually have two critical questions, because what you said finally means the end of nanomedicine, because one drug molecule is always more simple than a complex structure like a nanoparticle, which means that industry might not be interested in whatever we are doing here. The second problem is when you are going to personalized medicine or precision medicine, you are decreasing the number of patients, meaning you have a more specific drug. Also, this is against the interest of pharmaceutical industry because you don't create blockbusters. So what we are discussing on the conference has any meaning for the pharmaceutical industry? Sorry, very critical.
I certainly disagree with your statement. My point is that you need to understand that it's not straightforward to obtain new products for the market, whether they are nanoparticles or other drugs. And I hope one aim with such a presentation is that I warn some scientists that are working with nanoparticles that can never end the market. It isn't possible that they can reach the market. So they have to think about select a research area where you have that possibility. If you want to try to get to the market, if you are focusing on only basic research and animal studies, you don't need to think about that.
Okay, thank you very much. Then we have